All right, guys, welcome to another Fire in a Bottle video. Look at me. Look at the technical wizardry happening here. You can actually see me talk and you can see the slideshow at the same time. Have you ever seen this kind of wizardry before? Probably only on like every other YouTube channel ever, but I've figured it out. So, feeling pretty good about myself. Anywho, so I have a ridiculous video today called Cornflakes for Weight Loss. Yes, I'm serious. I've been trialing a low-fat, high-carbohydrate diet that includes the supplement SEA or steer oil ethanolamide, which I've been selling at Fire in a Bottle. You can find it at fireinaBottle.net/shop. And yes, often in the mornings, I actually eat cornflakes. I've also been eating a beef and bean chili that I like to make with grass-fed ground beef, which is one of my favorite dishes. And sometimes I just get lazy and eat pasta. Of course, this is a very easy diet to follow. I make the chili ahead of time. I keep it in jars in the fridge. If you followed my work at all, you probably realize that I'm doing this diet for a reason. Yeah, partially it's funny, but I think there are some fundamental truths about carbohydrates and redox balance that are super important. Lean and obese people tend to use carbohydrates differently, and that's what I'm going to talk about. We're going to focus on that a little bit, talk about why lean and obese people use carbohydrates differently, and then I'm going to talk briefly about why I think that SEA could be a key thing to help obese people use carbohydrates more effectively to enact the redox change that they want to be. Okay, so let's start off with John Speakman's paper. This paper shows the change in human metabolic rate since 1920 to the year 2020. And he's got a great thread about this on Twitter that I'll put in the comments below. He says, we are currently expending about 220 calories per day less for males and 122 calories per day less for females than people of our age and body composition were in the late 1980s. These changes are sufficient to explain the obesity epidemic in the USA. John Speakman is a really smart obesity researcher. He believes that the cause of the obesity epidemic could be a drop in metabolic rates, and I happen to agree with him. Of course, in parallel with dropping metabolic rates, we have seen decreasing human body temperatures since the Industrial Revolution. These high lines, so this is over the course of a human life. This is your body temperature at age 30. That's at age 80. The blue lines here, this is from the time period 1860 through 1940. And this orange line, which is about a degree Fahrenheit colder, those are measurements taken from 2007 to 2017. So human body temperatures have dropped by about a degree to go along with the fact that we're burning 200 calories less per day than we did 100 years ago. And the Speakman paper goes on to say it's not because of inactivity, it's because our basal metabolic rates have dropped. And if we get back to the starch for a minute, this is an article that I wrote on fireinaBottle.net called Pontzer's Burn and Metabolic Rate Mechanisms. If you go to fireinaBottle.net, click search and put in Pontzer, this will come right up. You can find the references to all these papers there, but what I like to point out is, so this is the metabolic rate adjusted per kilogram of fat-free mass of obese white males in this one study was found to be about 28.6. Now, if you compare that, this is Tsimane farmer foragers. This is a tribe that lives in Bolivia. They eat a lot of starch. They eat plantains, they eat manioc, and their metabolic rate is 38 kilocalories per kilogram of fat-free mass. These are Laotian male rice farmers. Their metabolic rate is almost 40. And female Thai rice farmers, in this study, they found the highest resting metabolic rate or basal metabolic rate that I have ever seen of 40.8 in these female Thai rice farmers. So what you see is that the 3 or 4 highest metabolic rates ever recorded were all recorded in starch eating cultures. And so that's why I'm interested in cornflakes. That first slide, I figured out if I eat cornflakes with 2% milk, that's about 73% of calories from carbohydrates. 90% of it is from starch. These are the female rice farmers that have the highest resting metabolic rates ever recorded. And you can see that their percentage of energy as carbohydrates is almost 84%. And so I'm talking about eating carbohydrates to burn fat. This is an old paper from 1936. I think it was published in 1937. And even then, this was an old saying, fat burns in the flame of carbohydrate. And I want to talk about why that is. Obviously, I'm eating starch, with the idea that I can use something about that starch to help burn my stored body fat. So this is a very oversimplified view of how we burn calories. This is fat and carbohydrates that come into our cells. We convert those fat and carbohydrates into something called acetyl-CoA. The fat and the carbs actually get made into the acetyl groups. The acetyl groups enter something called the TCA cycle, also known as the citric acid cycle, also known as the Krebs cycle. And to oxidize or to fully burn one acetyl group, which again comes from fat and carbs, what we need 3 molecules of NAD+, and we generate 3 molecules of NADH. And you can see this reaction happens 3 times. So the more NAD+ there is, the faster this reaction goes, the faster we burn calories, and the more NADH there is, this actually bogs this process down, and the slower we burn calories. So this is from a recent experiment. I think this is a 2020 paper called Acute Carbohydrate Overfeeding, a Redox model of insulin action and its impact on metabolic dysfunction in humans. So these are in humans. I'm going to focus right now on the open circles here. So these are lean humans. This chart essentially represents NAD+ availability in their mitochondria. This is their baseline level right here. And at time 0, they start eating bagels. And then over the next 2 hours, 120 minutes, they're going to eat these bagels and they're going to drink this glucose drink. I think it's equivalent of about 340 grams of carbohydrates, as I recall. What happens is almost immediately upon eating the bagels in the lean humans, the availability of NAD+ starts to rise. So by an hour in, and these people eating almost pure carbohydrates, almost pure glucose, they have a significant rise in NAD+, and that NAD+ stays elevated for around 2 hours. Now, conversely, this is a study done way back in 1970. They took these guinea pigs and they fed them either a starch-based diet, or in this group of guinea pigs, they just fed them olive oil for 3 days, right? And so they're forcing these guinea pigs to burn pure fat, and it's mostly unsaturated fat, or what it's going to be is actually a combination of the olive oil from the diet and a combination of their stored body fat. And this is the NAD+ availability. And so in the guinea pigs fed starch, you see NAD is up at 18, and in the ones fed olive oil, that number drops to 8. So in the mitochondria, when you feed guinea pigs olive oil for 3 days, the ratio of NAD+ to NADH drops by more than half. When you burn starch in the mitochondria, you can actually increase the NAD+ to NADH ratio. But when you're burning a lot of fat, especially unsaturated fat, in the mitochondria, you can actually drop the ratio of NAD+ to NADH, which, as I said, is the thing that makes your metabolism go, is that availability of NAD+. So this is all pretty straightforward, right? We just eat starch, right? But there's a problem. And so this is one of my favorite papers. This is from the Craig Tomlinson group. And what they did in this paper, so this is in mice, and they took mice and they put them, this green line is mice on a control diet. And so these guys are just eating a starch-based diet, a standard kind of chow or lab diet that is known to keep mice lean. These mice here in the black, these are on a quote-unquote Western diet. This is a pretty crappy diet. It's pretty high in fat. It's very high in unsaturated fats. And so they become obese, and over 20 weeks, they become very significantly obese. And then the third group is this red line. So in this red line, they put them on the Western diet for the first 10 weeks. They became somewhat obese, and then they switched them back to the control diet. So from weeks 10 on out, these mice in the red are on the same diet as the mice in the green. All of these mice are on the high starch controlled diet, but yet the mice in the red never return to the same body weight as the mice in the green. And looking on this graph, it looks like it may not be a huge difference. But if you look at these lines, the green is around 12 to 13 grams, and the red is maybe 16 or 17 grams. So you're talking about the difference between, if you put that in human terms, you're talking about the difference between a person who weighs 130 pounds versus 160 or 170 pounds on the same body frame, right? So this is really a massive difference. We're talking about a 30 or 40 pound difference in adult humans. And these mice are living on the exact same diet that kept these mice lean. But the problem is, being on that Western diet broke their metabolism, and just switching back to carbohydrates does not fix them, right? They're broken. And so what does a broken metabolism mean? What does it mean in these mice? The first problem is that the mice and also obese humans have too many unsaturated fats. And what I mean is your stored body fat. Because you may be thinking, well, if I eat all starch, why am I worried about how saturated the fat is? But of course, the fat that you have to burn is your stored body fat. And this is that same paper from John Speakman about how human metabolic rates have dropped. And one of the things they showed is that mice fed a diet high in saturated fat for their whole life have higher resting metabolic rates than mice who are fed unsaturated fats. And that is a really important distinction. Remember, those mice that had very low NADH were fed olive oil, right? They were fed fat that's mostly unsaturated. And of course, if you're trying to burn your own body fat, what you're working against is the saturation level of that stored body fat, especially if you're eating a starch-based diet. It doesn't matter what's in the food. It matters the fat that you're actually trying to burn, which is your belly fat and your back fat, right? I just wanted to point out how the composition of back fat has changed. So this is a study from 1962. I show the study all the time, but it's important, so I'm going to keep showing it. So in this paper, they went to different parts of the world. They looked at a tribe in Nigeria that was mostly eating starch. And what they found is their body fat was around 40% saturated, and it was about 50% monounsaturated, and it was about 10% polyunsaturated, very roughly. But this number is very useful to think about as a kind of human baseline for a starch-eating culture that is presumably going to have a high metabolic rate. This is another example of Colombia in 1962. They're a little bit less saturated. They're only at 35% saturation. And polyunsaturated fat in that culture was a little bit lower. They were down at 7%, and they had a little more monounsaturated fat. And then just to contrast that, so this is from Finland in 2010, and this is actually people getting laparoscopic surgery. So these are in humans who are very obese, and you can see that their saturated fat is all the way down to 27%. Their monounsaturated fat is up at 60%, and their polyunsaturated fat is up around 12 or 13%. And so the body fat of these people is just about as unsaturated as olive oil, as a lot of types of olive oil. And remember, you saw what happened when we fed guinea pigs olive oil for 3 days. And so if you're trying to burn a lot of your own stored body fat, you're going to have low NADH. Example, this is modern Americans. This is postmenopausal women done in 1998. Their saturated fat is down to 24%. Their monounsaturated fat is at 54%, and their polyunsaturated fat is way up to 21%. And just to show that I'm not totally crazy, these are French women in the year 2000, and the French women actually maintained their saturated body fat at around a third, and kept their monounsaturated fat to about half, and their polyunsaturated fat rose a little bit from those numbers we saw in the 1960s. The French have done a lot better eating butter and cheese at maintaining a kind of historically normal saturation level of human body fat. And the French to this day have the lowest obesity rates of citizens of any modern economy. So the second problem that the obese mice have who are not fixed is something called lipolysis. And this means lipo means fat and lysis means cutting or releasing. Lipolysis is the process by which your adipocytes, your fat tissue, releases fat to the rest of the body. They release free fatty acids into the bloodstream, and the other cells in your body can take up those fats and burn them for fuel. And so this is a classic paper, Effects of Obesity on Substrate Utilization During Exercise. And so one of the things you can see, so this is free fatty acids, and these open circles on the top are obese humans. And what you can see is they have higher levels of free fatty acids than the lean humans that they're compared to. Right. And so we can say that the obese humans are doing excessive lipolysis. Their fat cells are releasing more fat than is presumably necessary. And what that means is that when they start to do exercise, The lean humans actually burn more glucose. So this is respiratory exchange ratio. The higher you are on this scale, the more glucose you're burning and the less fat you're burning. So these lean humans, when they start to exercise, they burn glucose. And these obese humans, when they start to exercise, they're not able to switch over to glucose burning. They're stuck in fat burning mode. So we can say that one way in which obese humans lack metabolic flexibility is they're not able to switch over and burn glucose. They're actually stuck burning fat, which sounds ironic because if you're fat, you're probably thinking, I need to burn all of this fat. But remember that fat burns in the flame of carbohydrate, and you actually need that NAD plus provided by burning the glucose to help you burn the unsaturated body fat that you're stuck with. Here's just another example of this. This is from that same study I talked about at the beginning where people start eating bagels and they drink this glucose drink, and it's about 340 grams of carbohydrates that they consume. And again, you can see these are the lean humans here in the open circles, and they're burning more glucose. The obese humans are not able to switch over to burning glucose as efficiently as the lean humans do. And this graph just shows, so the lean humans have lower rates of free fatty acids to begin with. Once they eat, here's the meal, you can see that lipolysis drops pretty dramatically over the first hour. So one of the jobs of insulin is to decrease lipolysis. And so you can see when people eat, and in all cases, whether or not people were lean or obese, in all cases, once people eat a mixed meal, the amount of free fatty acids drop. So that means in all cases, everyone is reducing lipolysis in response to a meal. However, the lean humans are better at it. And so if you're worried that by stopping lipolysis in response to a starchy meal, you'll never burn fat again, the good news is that this process peaks at about 3 hours after you begin to digest those carbs. And then from 3 hours on out, lipolysis is increasing and you're burning steadily more and more and more fat. I've been trying to go 6 hours between meals. And since I'm not eating very much fat in the meal, by the time I get to the 3 hour mark, all of the fat that I consumed is gone. And over the next 3 hours, I'm burning my own body fat, but I'm doing it with the blood glucose that I got from those carbs and with the stored glycogen that I have. And so what I believe is happening is between the 3 and 6 hour mark, I am able to burn my own body fat in the flame of the carbohydrates that I've consumed. That is the theory. However, like the mice who are not fixed, I still have a problem because my curve of lipolysis and free fatty acids is going to look like the women with upper body obesity. I'm somewhat insulin resistant. I'm not going to be able to reduce my level of lipolysis equivalent to that of a lean person. And so I'm not going to get the same NAD plus benefit as a lean person out of the box. And so this study just shows exactly what I'm saying is that if you have high free fatty acids, and that's what's happening here, palmitate. So this is essentially the amount of free fatty acids in different humans. And as you go to the right on the scale, this means you're burning more carbs. So you can see people with low free fatty acids, like if you look around 75 on this and you follow it over, they're at a respiratory exchange ratio of about maybe 0.85 or 0.87. That means they're burning more carbs than they are fat. But if you go to people with a free fatty acids of around 100, you're down to an RER of about 0.8. And so if you have more circulating free fatty acids, you are going to burn more fat and you're going to burn less carbohydrates, and you're presumably going to have more NADH and less NAD+. This process has been described as the Randall cycle. What happens is you can see fatty acids are coming into the cell and into the mitochondria here, and they produce a lot of acetyl-CoA and they produce a lot of NADH. And glucose is coming into the cell over here, and it gets converted to pyruvate, and it goes into the mitochondria, and pyruvate dehydrogenase converts it into acetyl-CoA, and it goes into Krebs from there. But the problem is, see this red arrow with this square head, both acetyl-CoA and NADH, when they're high, block pyruvate dehydrogenase. And so when fat is high, fat comes into the cell, it gets oxidized, acetyl-CoA and NADH are high, and that prevents you from burning that glucose. And so that is why an obese person with high free fatty acids is not able to burn carbohydrates in the same way as a lean person with low free fatty acids. So again, to bring this back home and prove that I'm not just talking smack, I already showed this, but I want to just focus again now on the obese humans. And so I talked before about the lean humans. So again, these are the people who start eating bagels and glucose at time zero. In over 2 hours, in the lean humans, you see this immediate jump in NAD plus levels, right? And they're generating NAD plus and they're burning calories. But in the obese humans, you do not get that same jump in NAD plus levels. In fact, at first, you get a drop. Eventually, the obese humans start to be able to burn some carbohydrates, and these NAD plus levels do rise a bit. But obviously, over the time course of this thing, the obese humans never have the same amount of NAD plus as the lean humans. And I'm arguing that one of the main reasons, if not the reason for this, the higher levels of very unsaturated free fatty acids in the obese humans are taking priority of oxidation via the Randall cycle, and they're essentially inhibiting the ability in the obese humans to burn glucose. Now, an interesting thing about this is we make something called tissue necrosis factor alpha. TNF alpha is considered to be a pro-inflammatory cytokine or a pro-inflammatory mediator. In this study, they looked at adipose tissue, and they showed that in lean humans, that's the open bar here, levels of TNF alpha are at about 25. And in obese humans, levels of TNF alpha were almost 60. And so obese humans have significantly elevated levels of TNF alpha. Now, that is very interesting because if you take adipocytes in tissue culture, if you take fat cells and you give them TNF alpha, what will happen is you'll have a huge increase in lipolysis. So these are adipose cells growing in tissue culture for 18 hours. The white bars without TNF alpha, the black circles are with TNF alpha. What you can see is there's a literal tripling of lipolysis in adipocytes that are cultured with TNF alpha. Furthermore, you can see that the more TNF alpha there is, the higher people's plasma glucose is, and that's presumably because if you have more TNF alpha, you're doing more lipolysis, you're blocking the ability to burn that glucose and get going. And so this is, again, that same study from the Tomlinson lab that showed the mice that were not fixed. And so what they did is they used this pharmaceutical inhibitor of something called the aryl hydrocarbon receptor. And if you want to learn what that does, look at my earlier videos in the Obesity Explained series. The first 8 or so videos are all about the AHR and all of the ways that it is affecting your NAD plus levels, really. So anyway, what they showed is that by blocking the AHR and restoring NAD plus levels, you can actually fix the mice in a way that you can't do just by switching them onto that high starch diet. As I said in the beginning, I've been trialing something called steer oil ethanolamine, also known as SEA, also known in this experiment as N-steer oil ethanolamine, or NSE. If you want to try some of this, I do have it for sale at fireinabottle.net/shop. What they show in the study, these are rats that they made insulin resistant. In the insulin resistant state, their levels of TNF alpha triple, which is, remember, very similar to the difference between obese and lean humans of TNF alpha in their adipose tissue. This is unfortunately in plasma. They didn't look specifically at adipose tissue. And what they show is that in insulin-resistant rats, given SEA, the levels of TNF alpha return to control levels. And that's very impressive. And they showed in another paper that SEA actually binds to NF-kappa beta, which is the kind of OG transcription factor that controls a lot of inflammatory and lipogenic processes. And it actually stops that NF-kappa beta from translocating into the nucleus and producing tissue necrosis factor alpha, which is one of its target genes. This HOMA-IR is an insulin resistance score. A high score is bad. The insulin resistant were at 1.34, and you gave them SEA, their level of insulin resistance drops down to 0.75. So that's nearly half as much. So the rats given SEA of something like half the level of insulin resistance as the normal insulin resistant rats. What I see as some of the issues being is that metabolic rates have dropped. This is in large part due to the consumption of unsaturated fatty acids. If we look at high cultures, they have very high basal energy expenditure or very high resting energy expenditure, which is exactly what Americans no longer have, right? Americans used to have high resting energy expenditure and be lean. And now to find humans like that, you have to look at starch eating cultures. But the caveat in all of this, as we saw in the mouse study, is that switching to starch may not fix you once you're broken. And I talked a lot about the reasons for that. Part of that is that you're full of very unsaturated fat that are not going to regenerate NAD+. And the other reason is that you may be doing inappropriate amounts of lipolysis. You may have high levels of circulating fats that you don't want if you want to be able to burn glucose and get that NAD+ spike from efficiently burning glucose so that you can burn your stored body fat in the flame of carbohydrate. With all of this in mind, I am doing a literal cornflake trial. I'm not a Thai rice farmer. I'm American. I like cornflakes. I grew up on cornflakes, but this is the American version of a starch-based diet. I'm doing 2 meals a day, typically about 6 hours apart. As I say, I think that second half, that time from 3 to 6 hours, that's when my body is full of glycogen. I have carbohydrates available to burn, and I have steadily rising lipolysis. So I'm steadily burning more and more and more fat as I stretch that time between those 2 meals. For me, it's not a problem to have a relatively large starch-based meal and then go 6 hours before the next one. My daily caloric consumption has been on days that I've only calculated about 3 days so far. It's been between about 2,200 and 3,300 calories. The 3,300 calorie day was yesterday. I was hungry. I played basketball the night before, and I wound up consuming more calories. But I'd say more typically, I've probably been averaging around 25 or 2,600 calories. And as I say, I'm taking 600 milligrams of SEA every morning, first thing, to control that tissue necrosis factor alpha and to control that unnecessary lipolysis that is increasing my free fatty acids and making me insulin resistant, making me resistant to burning that glucose in my diet. The last part of this plan, and if you watch my videos and look at my blog, I talk about these things a lot. I am using some oxidants. I'm using R-alpha lipoic acid. R-alpha lipoic acid is mitochondrial specific. It converts NADH to NAD+, and so it lowers that ratio of NADH to NAD+. AKG, I've talked about quite a bit. That's another one I sell at fireinabottle.net/shop. AKG helps you to break down branched chain amino acids, and it's also highly thermogenic, and it also drives NAD+ production. Lastly, disodium succinate, I also sell it at fireinabottle.net/shop, is another one that you can use to help push up those levels of NAD+. I hope that makes sense, guys. I am trying to use a traditional starch-based diet to get my metabolism going. And the SEA and the oxidants are the tools that I'm using to help me get unstuck, to help knock down that lipolysis, to help generate NAD+, so that I can stop burning extra fat that I don't need to burn right now, and so that I can burn more carbohydrates, and so that I can burn my stored body fat using the NAD+ advantage that those carbohydrates give me. To this point, the advantages that I've noticed, and I talked about this in the last video, I have had steadily elevated body temperatures. My body temperature when I first wake up has been around 97.5. I have the SEA right away. I've typically been eating about 1,000 calories of cornflakes and milk around 9:30 or 10 a.m. Then most of the day after that, my body temperature is between 98.5 and about 98.8, and it'll stay that way until maybe 8 or 9 p.m. or 10 p.m. The body temperature will start to drop, an hour before my sort of normal bedtime, which is normal. You have to lower your body temperature back down to sleep. And I am losing weight. It's too early to sort of spike the ball in the end zone. But I'm down about 7 or 8 pounds over the course of 2.5 weeks, and 6 days of that or so were over July 4th, when I was not on any kind of regular eating schedule and I was eating a lot of junk food at my family's place. And so I'm seeing good progress. even in the face of that. This is a very easy eating plan to follow. The SEA, in addition to the metabolic benefits, does provide some appetite suppression. Like I say, I'm still consuming probably 2,600 calories a day on average, so it's not like I'm starving myself. But the SEA helps with just hunger pangs and that kind of sometimes you just are feeling snacky, and the SEA seems to help with those kind of impulses, I've noticed. Okay, guys, that's all I've got for now. I hope this helped. I hope this has helped make some sense of my last video when I said that I wanted to try a more thermogenic meal, and so I ate a bowl of cornflakes. Some of you had questions about that. I'm sure there'll be more questions. Ask them down below in the comment thread. I'm trying really hard to keep up with comments and questions. Also, I know some of you have had the SEA for a while now. I'm curious about your experiences, about your thoughts. You can leave those in the comments down below. As I said in the last video, I don't recommend more than 1 capsule of the SEA a day. I was doing 1,200 capsules for a while, and I nearly overheated. My body temperature went over 100, and I started almost having flu-like symptoms the first day that I went to a very high-starch, low-fat diet. And so I would say take your time, take it slow. 1 capsule of SEA a day is probably plenty for most people. Okay, so that's where I am with cornflakes. Have a great week, guys. I'll see you next time.